Good morning, everyone, and welcome to Scintica's morning coffee chat. My name is Jade, and I'm a product manager here at Scintica for our X-ray and optical imaging portfolios. And I'm here this morning with my colleague, Sadi.
Good morning, everyone. My name is Sadi, and I'm a product manager for our ultrasound portfolio. And today we will be discussing some of the differences between ultrasound and DEXA and how they can be combined in your research.
Let's start by comparing the two modalities at the core of this conversation. Does DEXA provide a more accurate assessment of metabolic changes than ultrasound-based fat and muscle quantification?
So what I would say is that with DEXA, we're going to be able to get a really highly accurate representation of muscle and lean mass, just based on the fact that we're able to get total body quantification measurements as well as user-defined regions of interest. So we're able to use an unlimited number of them with this particular DEXA system. So you're able to look at, again, whole body mass and then in one specific area. And because the Insight DEXA has been fully validated against other gold standard methods for measuring body and bone composition, we're able to, again, have lots of confidence and know that with high reproducibility, the data is going to be right each time. The other thing that I want to mention as well is that because it's a really low dose radiation system, you're able to do repeat scans over a longitudinal period of time for your study without this fear of the negative sort of biological side effects that can happen with other higher radiation systems. So again, you're not impacting or confounding your experiments by using this system. And then, of course, because it's a DEXA system, we're placing the animal inside of the scanner. There really isn't any sort of user bias involved. And so kind of researcher to researcher, you're not impacting kind of the analysis, or sorry, I guess the quantification and measurement of your animal. So from that perspective, again, you're getting highly validated, highly reproducible measurements without having to worry about how one researcher measures versus another. It's right every time.
All right. So when it comes to ultrasound and metabolic diseases like obesity and diabetes, the great part with ultrasound is that it's very universal. Most people use it for looking at cardiac or liver or other abdominal issues that abdominal organs that might be affected in these diseases. One of the smaller areas that ultrasound isn't really known for is when it comes to muscle and fat imaging. But the great part with ultrasound is that you can use it to look at regional fat, and you can even use it to look at muscle thickness. So while it cannot complete full body analysis at the same time or full body imaging, you can distinguish features like visceral fat and subcutaneous fat, and that can be really relevant in specific metabolic diseases like obesity. Another great feature I like about ultrasound is that it's completely non-ionizing. You can image your animals or even in clinical studies as many times as you want without worrying about any side effects. And you have the capabilities of going into more advanced imaging, such as 3D imaging. You can incorporate pulse wave Doppler and color Doppler to look at blood flow in these areas. And you can even start to get more functional data with elastography and stiffness and really advance your studies into the next level.
Let's continue by examining how these two modalities perform when it comes to measuring body composition. Is DEXA still the gold standard for quantifying bone and fat mass, or can ultrasound achieve comparable accuracy with greater versatility?
So that's a great question, because I get asked all the time when we're showcasing the ultrasound system, can we look at bones and can we look at bone mass and fat mass, which, as everyone knows, it's not ultrasound's strong suit. Ultrasound is really good for soft tissues. It's really good for cardiac and abdominal tissues. But there's a lot of advancements that are currently being made in order to take ultrasound in that direction. So when we're talking about bone and body composition and we want to look at bone quality, ultrasound can be used to look at bones, both preclinical and clinically. If you go with a high frequency system, you can get down to about 30 microns and look at bone fractures. You can look at disc relocations in the spinal cord, and you can really get creative depending on the positioning of the ultrasound system. One of the nice features with ultrasound is that it does offer real-time imaging. So if you do want to do dynamic studies, if you want to look at bone quality alongside with blood flow, for example, in the spinal arteries and the spinal cord, you have that option. It's a very portable system, so you have the flexibility to reposition your animals or patients in any position that allows you to enhance image quality. And ultrasound is coming up with a lot of new methods to look at bones. For example, shear wave elastography is one of the newer modalities being introduced preclinically and clinically. And it's a way of not getting nice images of bones, but you can also get quantifying elastogram maps where you can look at bone stiffness and get more functional data.
So I think with DEXA, there's a number of different ways to measure body composition, bone composition, both clinically and preclinically. And so DEXA is going to be one of those gold standards that is a preferred tool in healthcare for getting kind of a non-invasive, quick assessment of what's happening. And so preclinically, what we see a lot is DEXA use as well as echo MRI. And as we heard from Sadie there are some ways that we can use echo or ultrasound to get information about body composition as well as bone. But in terms of DEXA, it is a very standardized and very well validated modality for looking at this. So lab to lab protocols exist, guidelines to how to use these scanners exist. And it's really easy to, perform your experiments in one lab, move that system to a different lab, scan those same animals again, and get very similar data. So again, it is standardized and validated. So the reproducibility and ability to kind of repeat scans over time and know that really the only thing changing is whatever your experimental intervention is, that's what you're hoping to get. With the Insight DEXA, from a service standpoint and general support, we do use daily test phantoms at the beginning of every day whenever you're going to start your experiments for the day. And so that the system is working up to par and it's fully working within spec. So again, the accuracy of that system and its performance is there. We also do preventative maintenance work with these systems in the service contracts that we offer, which means that we'll come on site and perform a full factory calibration, again, to make sure that everything from an accuracy and precision standpoint is working properly. So again, you can be using DEXA to kind of get a general sense of whole body physiology and anatomy, but also it can be used as kind of a first pass to determine which animals you want to send to maybe a higher radiation type of technique, so micro CT, where there would be a very firm endpoint, or perhaps to kind of decide which animals to send again to the point where you'd be now working more with ex vivo or in vitro modalities. It can be kind of a nice checkpoint type of technique as well.
So, Jade, I have a question then. With the DEXA, is it more popularly used for bone imaging or is it more popularly used for body composition? And if people who are interested in bone, they want to get more high resolution, do they then have to go to micro CT, or is DEXA advancing in a way where you can actually get high resolution and get the body composition all in one go?
Okay, excellent question. So what I would say is of the 35 plus sites or install bases that we have across North America and globally, I want to say that we actually have a pretty good split. So there's a fair number of researchers who are kind of all bone or orthopedics, and then there's another body that is very much kind of metabolism, your more traditional body composition of fat and lean mass. But what I'm seeing in terms of a trend, and we can talk about this more in a moment, is with kind of this movement towards developing weight loss drugs, and we're trying to, with certain pharmaceuticals out there, we're trying to lower the fat content, and we're seeing the impact that might be having on lean tissue. There is also a certain component where the bones are being affected as well. And so we're starting to see maybe where there used to be two schools of very much bone and then body composition, they're sort of blending together more so now, because we see, in terms of whole body physiology, integration of systems, they impact one another. So I would say that we're definitely moving towards a blend. And then, of course, for researchers who are still strictly bone, and that's kind of their plans going forward, there is a high resolution option on the DEXA. So our digital radiography mode, whenever you purchase the system, you can choose this add-on, which gives you higher magnification and higher resolution X-ray images. Now, with that, you're not able to get body composition images and data at a higher resolution and magnification. But if you're looking for nice representative images for your publications, you can choose the DR mode package and get nice, crisp, representative images for your figures.
Continuing our discussion on imaging in translational research, which modality offers more predictive value for therapeutic outcomes in translational studies, ultrasound or DEXA?
Okay, so for this question, this is where I truly feel ultrasound is the most superior when it compares to DEXA imaging or other modalities. So the real benefit of preclinical ultrasound imaging is that it truly mirrors clinical ultrasound imaging. The organs that you're imaging in a mouse or a rat or a rabbit, for example, you can apply those exact same techniques when looking at humans. The parameters that you're looking at, for example, if you're looking at liver, muscle, cardiac, any type of therapeutics and disease progression, you can easily translate that over to clinical studies, which is what makes ultrasound one of the best modalities used in preclinical and clinical research. The nice part about ultrasound as well is that you're not just limited to structural images, but you can get both structure and function. You can look at specific areas in the body, or you can look at the entire body globally, with different features like perfusion imaging, vascular, contrast, and shear wave. You have all of these different modes available at your fingertips. And with our Prospect T2 preclinical ultrasound system, all of those modes are integrated in the basic package. So with just one system, you have an open platform to perform many different types of experiments, gather lots of different types of data, both visual data and quantifiable data, and you can do it in real time in longitudinal studies. You can image your animals on a weekly basis, on a monthly basis, up to the entire duration of the study, and you can incorporate all of these different experiments and all of these data sets into one comprehensive study.
I think the conversation around improving translational research, sort of what we get in preclinical studies, and making it relevant or applicable to healthcare that's human related or in the clinic, is perhaps less of a question of the equipment or the modality, but also more so in sort of the animal model. So one of the things that we at Scintica offer, in addition to just kind of the equipment, is sort of the scientific expertise. So as customers, you're developing animal models for using any piece of our equipment that we offer. We're there to kind of support you and make suggestions and recommendations so that your animal model can be more human and clinically relevant. When it comes to the DEXA, the measurements that you're getting out of it, kind of with like a one-click sort of automation, you basically get all of your information about body and bone composition right away. The total mass, the fat percentage, the lean mass, lean percentage, and bone information, those are the same measurements that you would be getting in the clinic. Now, the Insight DEXA that we sell, it is manufactured by a company called Osteosis. And so they are very much in the DEXA body composition analyzer space, where they began in the clinic developing those systems for human use, and more recently, over the past 5 or 6 years, moved into the preclinical space. And so whenever it comes to the Insight DEXA for in vivo preclinical stuff, for mice, rats, non-rodents, tissues, bones, there's a number of different species that can be selected in the software so that specific algorithms can be applied to the scans, whatever the detector is picking up, so that it's giving you the most accurate results for the species that you're using. So again, within a certain animal model, you can be certain that the information that is being acquired and then you're analyzing is as accurate and precise as possible, so that the information that you're getting about your studies, whether it be a pharmaceutical that you're looking at, a certain bone disease, is giving you accurate information in the preclinical space, so that as you're then using other orthogonal approaches to build your studies and publish your literature, that is then going to be helping to translate in years to come into the clinical space for humans.
So Jade, would you say, because I'm not as experienced in DEXA, I'm curious, with the clinical side of DEXA, is there any advantages or disadvantages with the preclinical and the clinical systems in terms of how they acquire data? Or is it completely translatable in terms of you can reproduce everything from a preclinical model to a clinical model?
It's less about the technology being the predictive factor. The technology is doing what you're telling it to do. And then sort of by using different modalities, let's say if you're trying to combine DEXA and ultrasound, you should be getting the same trends. It's not that you would now be getting contradictive data or results, so that whenever you're then shifting into larger animals. So again, if you're talking about the drug development or the drug pipeline process of beginning in vitro or some kind of in silico stuff, the drug discovery stuff, into preclinical small animal, into preclinical larger animal, into the first stage of human trials, which would be all about safety before efficacy, and that stuff going forward. Like, you just want to make sure that whatever you're expecting that kind of preclinical equipment to tell you, like, that is what it's doing. And so you can combine different techniques to kind of all support one another. So whenever you start to see that something is off, or now the ultrasound is saying fat mass is going down, whereas your DEXA is going up, like, that causes you to stop it and think about it. Think about your animal model. Maybe it's not necessarily that your equipment is malfunctioning so much that the way that you're building your animal model or your breeding colony, something is changing. So that's kind of one of the challenges with preclinical research. And there's advantages to doing purely human research versus purely animal stuff. But again, it kind of speaks to how important it is that as we work towards new approach methodologies and away from such a heavy reliance on animal use, that everything kind of supports one another. So it's kind of working with the researchers to set up experiments that make sense and are well designed and robust.
See, no, that's a great, the point I like that you mentioned is refining the animal model specifically, because the one thing I run into a lot is when we do a lot of ultrasound studies and ultrasound demos, a lot of the professors I meet are also clinicians, and they do clinical ultrasound studies. And the main differences between, even though preclinical and clinical ultrasound work in the same physics and they acquire data in the same way, the biggest difference is the frequency. In order to do preclinical ultrasound studies, we operate at a very high frequency, which gives us a very high resolution. But because of that, we can't go as deep into the body. Now, with that, we say when we're looking at animals, we can pick up very fine details and very fine structures. One of the concerns could be when we transition this over to clinical studies, is a clinical ultrasound capable of picking up the exact same details and resolution? Because now we're dropping the frequency, we can go much deeper in the body, and we have to because humans are much bigger, but can we still pick up the same level of detail? And I think that's where you mentioned when you're designing these studies, you really have to factor in, when I'm translating from one species to another, will my equipment transition over? And can I transition over the same data sets and the same trends?
And I mean, again, like I was mentioning, we have a good number of users now across North America, a very established user base, which means, we're in government facilities, we are in biotech, we're in pharma companies, research hospitals. And with that, it's funny speaking to researchers who are now using a preclinical DEXA for the very first time. And previously they were using human DEXAs to scan their animals, which again, don't have the algorithms optimized for that species. So it can be a bit of a shift into now kind of re-optimizing, getting more accurate results whenever we're using a preclinical system that's, again, designed for small animals, that now you're getting accurate, reproducible information each time. So it's great because we're able to now improve the translatability of a lot of the research that's going forward.
Continuing our discussion, our next question is, should preclinical imaging prioritize non-ionizing ultrasound over radiation-based DEXA to improve welfare and reproducibility?
So on that point, this is one of the main key features of ultrasound that everyone loves and is often brought up over and over again in both preclinical and clinical, is that ultrasound is completely non-invasive and it's completely non-ionizing and radiation-free. So if you have studies where you want to image your animals or your patients on a weekly basis or a daily basis, there's absolutely no problem with that. If you are focusing on reducing your sample sizes and your animal cohorts because you want to have very specific targets, you want to make sure you're not using more animals than you need to. With ultrasound, it gives you that flexibility where you can image multiple organs, multiple tissues. There's no real limitation on what can be used with the ultrasound system, because whether you're looking at perfusion or blood flow or cardiac or abdominal, all of it is completely non-invasive and non-ionizing. And the nice part, which we've talked about in the past, is that with ultrasound, unlike other modalities, you can use it for both structural and functional measurements. You don't necessarily have to use contrast agents if you don't want to, but you can still get data like perfusion and stiffness. You don't have to expose the animal to any radiation or expose the patient. So this is one of the key areas where I think ultrasound really excels.
So I would say not necessarily. A lot of the first time sort of customer calls that we'll have are users who have previously been using CT or micro CT. So whenever they learn about the low dose that comes with our Insight DEXA, they're often very surprised and very excited. So based on kind of what's been established as the upper threshold for radiation levels that are biologically safe for repeat scanning, the Insight DEXA comes nowhere close to that, meaning that you could honestly scan several hundred scans a day to a mouse and still not see any kind of negative biological side effects. So from that standpoint, it's quite negligible, the radiation aspect. Also, with the Insight DEXA, it has a very fast acquisition scan time. So it's about 25 seconds. So it's 5 seconds of the low dose radiation and then 5 for the high, and then it's 15 seconds of post-image processing, which means that the amount of time that the animal is spending under anesthesia and how much stress is kind of being imposed, it's dramatically reduced. Now, also because with the Insight, we're getting really reproducible and accurate data, it kind of lowers the reliance that we will have on having to do repeat scanning. So sometimes with other modalities, you might choose to do your scans or acquisition in duplicate or triplicate. With the Insight DEXA, you really only need one scan, so long as the animal is placed nice and still. One is all that you need. And then the last thing that I'll mention again is that it is very ethically accepted standard approach for looking at body and bone composition. So it's not terribly difficult for getting it approved on any of your animal utilization protocols. It's also fully self-shielded, which means that it can be positioned in any lab space without any sort of additional radiation protection around it. So both user and animals are safe.
As we wrap up today's discussion, let's step back and consider how these imaging modalities might work together. Does combining ultrasound and DEXA enhance data interpretation or add unnecessary complexity and cost?
So I will say ultrasound is one of the founding imaging techniques, so to speak. It's been around for ages. It's been used for decades, and it's well known in the field of medical imaging. In the past, medical imaging, when it was very expensive and people can't do multimodal imaging, people were using clinical systems and preclinical studies, and things weren't optimized. We were limited. And in that scenario, ultrasound was one of the key systems used because you can almost image everything in the body. It's a very collaborative tool that you can use with lots of collaborators and professors and clinicians. And regardless of which disease you're focusing on, ultrasound was a great device to incorporate. So with ultrasound, the nice part about it is it's low cost. We have these nice compact preclinical systems. For example, the Prospect T2 can be set up right on your workbench. You don't have to remodel your animal room, reconfigure any space. You can set it up and move it wherever you need to be. It simplifies your workflow. It's easy for handling. You get lots of different parameters incorporated, lots of different features, though there is a lot of technique with ultrasound. Ultrasound is very manual. There is a lot of expertise that is required in order to operate it. And the great part with that is we take care of that for you. So because we have amazing application specialists at Scintica that can teach you different application techniques, regardless if you're imaging the heart, the muscle, the liver, if you're imaging everything, we can teach you all the necessary techniques in order to really enhance your studies. But at the end of the day, there are things where ultrasound will be limited to. And this is where Scintica as a whole, because we have our preclinical imaging portfolio, systems can be integrated and they can be combined all together, and they can be used all within one study to really take your studies to the next level.
So I think we're totally in agreement on this. Whenever it comes to, what ultrasound can do, what DEXA can do, every system has its strengths as well as therefore its weaknesses, because it's not necessarily designed to do that, which is where orthogonal approaches in science are so important, especially as we're seeing research studies and the way that published papers are kind of being presented now. There's multiple tiers and levels of experiments that have to be combined to give you to see a big picture of what's happening. As you mentioned there with Scintica, the way that we are kind of structured is that we offer a number of different imaging modalities, which, we've got some customers where they've got multiple of our systems either in their lab or across their cores, because they can truly appreciate the additional application scientific support that we offer, as well as the high quality technology that we sell. And so in that case, we've seen instances where the ultrasound system, the DEXA system are in the same facility, but they may or may not be being used for the same experiments. They may just be different groups that are using equipment sort of in parallel with one another. So as we talked about there, with ultrasound, there is a certain amount of perhaps user bias that can kind of come into play. So it kind of emphasizes the importance of being well trained in how to do that modality. With DEXA, it can be perhaps a little bit more of an unbiased approach to looking, again, primarily at body and bone composition. And in that regard, it can provide some complementary quantitative and qualitative information. So, structurally, you get a lot of really great information with ultrasound. Structurally, we can get similar information because we're getting a high resolution X-ray with our DEXA system. So it's not just body composition, bone composition. You are getting a nice X-ray image. And with that, with unlimited regions of interest that you can apply, you can look at length, and so how big or small certain features in the anatomy they are, as well as information about the composition and the amount, I guess, of body or bone and lean or fat mass. And so I think collectively we can kind of all agree at Scintica, the way that we sort of present our portfolio is very much how they can all complement one another. It's never that one is necessarily superior to the other, but just kind of how we can best support your research. We're here kind of on your team.
And this is where I think I really appreciate the multimodal approach that's been developing recently in the preclinical research space. There's a lot more emphasis on not just multimodal research, but multimodality research, in the sense that you want your individual systems to be able to acquire multiple different parameters, whether it's ultrasound where you can get structural and functional data, or DEXA where you can get bone data and body composition data. But with multimodality setups, like Jade mentioned, in one room, you can have an ultrasound, a DEXA, an optical, and an MRI. Because these systems are so compact and because they don't interfere with one another, we've done studies where an animal can be placed in the DEXA. After 30 seconds, while it's still under anesthesia, you can just move them over to the ultrasound, and then you can do your ultrasound imaging. And then within under an hour, you can get data from 3 different imaging systems, and then it can all be integrated with specific imaging softwares. And that's one of the real, I think, nice features that I like about our new imaging strategies.
Mm-hmm. And one final point to all of that, it can sound perhaps a little overwhelming trying to imagine acquiring all of these large pieces of equipment into your lab. Again, which is why there are other alternatives to using this equipment, whether it's through, CROs that we can link you up with, just kind of getting demos, rentals, etc. We are open to the conversation with customers to kind of figure out what the best strategy is to get you the data that you need as fast as possible. Because, in some regards, science can take many years, but as we've seen over the last couple, it's moving very quickly as we're trying to advance healthcare and human research. So again, coming with very complementary, high quality pieces of equipment to help you achieve those results, Scintica is kind of here to support you.
So we'd like to thank everyone for listening to our morning chat today. And we hope that you learned something new about the ultrasound system or the DEXA system and what Scintica offers as a whole. If you have any questions, make sure to click on the link below. You can always send us meeting requests where we would be happy to meet with you, learn about your research, learn about any of the challenges you're facing and how Scintica can support you in your future research.